Hello everyone, welcome back to the channel Microbial World. Antibiotics are one of the most powerful discoveries in medicine. But have you ever wondered how they work inside our bodies to fight bacteria? In this video, we are going to break down the mechanism of action of antibiotics in an easy way, from how they block cell wall formation to how they stop protein synthesis. So let's get started. Antibiotics are substances that kill or inhibit the growth of bacteria. Bactericidal antibiotics kills the bacteria directly, whereas bacteriostatic antibiotics do not kill the bacteria immediately but stops their growth or reproduction. Antibiotics target specific bacterial structures or processes that are absent or different in human cells. The main targets of antibiotics includes cell wall, ribosomes, nucleic acids, metabolic pathways, and cell membrane. So antibiotics can be classified based on how they act against bacteria. As we said, some antibiotics destroy the bacterial cell wall. Examples are penicillins, cephalosporins, vancomycin, bacitracin. Some other antibiotics blocks the protein synthesis like tetracyclines, aminoglycosides, macrolides, chloramphenicol. Antibiotics that inhibit nucleic acid synthesis includes quinolones and rifampicin. Some antibiotics can also interfere with the metabolism like sulfonamides and trimethoprim. Very few antibiotics damage the cell membrane, such as polymyxins and daptomycin. Inhibition of cell wall synthesis. One of the most important targets of antibiotics is the bacterial cell wall, which is made of a unique molecule called peptidoglycan. Now, human cells do not have a cell wall, so this makes it an excellent target for selective toxicity. Beta-lactam antibiotics such as penicillin, cephalosporins and carbapenems work by binding to enzymes known as penicillin-binding proteins. These penicillin-binding proteins are essential for cross-linking the peptidoglycan strands, which provides strength and rigidity to the bacterial cell wall. By inhibiting these enzymes, beta-lactams prevents the formation of a proper cell wall, leading to cell lysis due to osmotic pressure. Another antibiotic, vancomycin, also works by inhibiting cell wall synthesis but through a different mechanism. In bacteria, the peptidoglycan cell wall is made up of sugar chains, that is NAM and NAG, N-acetyl-muramic acid and N-acetyl-glucosamine linked to short peptide chains. At the end of these peptide chains, there are two D-alanine amino acids joined together. This is called D-alanine terminal. Vancomycin binds to the D-alanine terminal of peptidoglycan precursors, preventing them from being incorporated into the cell wall. So it prevents cell wall polymerization and causes bacterial death. As the inhibition of cell wall synthesis leads to bacterial lysis, it is considered as a bactericidal mechanism. Inhibition of protein synthesis. Bacteria have 70 S ribosomes, which is structurally different from 80 S ribosomes found in human cells. Antibiotics exploit this difference by binding to specific subunits of the bacterial ribosome and interfering with protein synthesis. Aminoglycosides like gentamicin and streptomycin bind to the 30S subunit of bacterial ribosome. This causes the ribosome to misread the mRNA code, resulting in the production of faulty proteins and leading to the death of bacteria. So there is a bactericidal effect. Tetracyclines also target the 30S subunit, but their mechanism is different. They block the attachment of amino-acyl tRNA to the bacterial ribosome, thus preventing the addition of new amino acids to the growing peptide chain. This halts protein synthesis and it has a bacteriostatic effect. Macrolides such as erythromycin and azithromycin binds to the 50S subunit of bacterial ribosome. They inhibit the translocation step where ribosome moves along the mRNA. This stops protein elongation and inhibits the growth of bacteria. Chloramphenicol also targets the 50S subunit of bacterial ribosome. They inhibit the peptidyl transferase enzyme, which is essential for forming peptide bonds between amino acids. The mechanism of clindamycin is similar to that of chloramphenicol. So by disrupting protein synthesis, these antibiotics either kill the bacteria directly or inhibit their ability to grow and multiply. Inhibition of nucleic acid synthesis. Certain antibiotics target bacterial DNA replication and RNA transcription processes. Fluoroquinolones such as ciprofloxacin and levofloxacin inhibits enzymes called DNA gyrase and topoisomerase 4. These enzymes are critical for supercoiling and untangling bacterial DNA during replication. Inhibition of these enzymes leads to DNA damage and prevents bacterial multiplication, leading to bactericidal effect. Rifampicin works by binding to the beta subunit of RNA polymerase, the enzyme responsible for synthesizing RNA from DNA. By blocking this enzyme, rifampicin prevents transcription, stopping RNA and protein production, which eventually kills the bacteria. So these antibiotics interfere with the genetic machinery of the bacteria, stopping cell division and leading to bacterial death. Inhibition of metabolic pathway. Some antibiotics target the metabolic pathways, particularly the synthesis of folic acid. Folic acid is essential for DNA synthesis and bacterial growth, but the bacteria must synthesize it themselves while humans obtain it through diet. Sulfonamides act as competitive inhibitors of the enzyme dihydroapterate synthase, which is involved in the early steps of folic acid synthesis. This blocks the formation of dihydrofolic acid. Trimethoprim, on the other hand, inhibits the enzyme dihydrofolate reductase, which is required to convert dihydrofolic acid to tetrahydrofolic acid, the active form used in DNA synthesis. When used in combination as cotrimoxazole, these two antibiotics blocks two consecutive steps in folic acid pathway, resulting in a synergistic bactericidal effect even though individually they are bacteriostatic. Disruption of cell membrane integrity. Some antibiotics directly damage the bacterial cell membrane, leading to the leakage of essential ions and molecules, which causes bacterial cell death. Polymyxins such as polymyxin B and polymyxin E binds to lipopolysaccharides and phospholipids in the outer membrane of gram-negative bacteria. This interaction disrupts the membrane structure, increasing its permeability and leading to cell lysis. Daptomycin, used against gram-positive bacteria, inserts itself into the bacterial cytoplasmic membrane in a calcium-dependent manner. It forms pores in the membrane, causing the leakage of potassium ions and depolarization of membrane potential, which quickly results in bacterial death. So this mechanism is rapidly bactericidal. So to conclude, in today's lesson we learned that antibiotics kill or inhibit bacteria by targeting essential bacterial structures and functions such as the cell wall, ribosomes, DNA and RNA synthesis, metabolic pathways, and cell membrane. Understanding these mechanisms is crucial for using antibiotics effectively and preventing the rise of antibiotic resistance. I hope that the video was useful for everyone. Please do like and subscribe the channel for more informative videos. Thank you.